Hello and welcome to a quick video on microtubules, one of the filaments that make up the cytoskeleton of cells. Microtubules are formed from long hollow cylinders of tubulin that are about 25 nanometers in diameter and comprise of tubulin monomers made up of alpha and beta tubulin subunits. The monomer is therefore described as a heterodimer. Like actin, microtubules have a positive growing end, but unlike actin, microtubules are not branched. Microtubules determine the position of organelles in the cell and are also involved in intracellular transport. The microtubules are formed as a star array which radiates out from a single center consisting of a centrosome, also referred to as the microtubule organizing center, which is located near the nucleus. Microtubules also form the mitotic spindle and provide the protein core of cilia and flagella. Overall, the microtubules are more rigid than actin filaments. Although the microtubule is a rigid structure, the overall filament is unstable in that it is in a state of flux. That is, the two subunits can rapidly depolymerize and the structure collapse. And this control of stability is determined by the enzymatic activity of the heterodimer. Now, this brings us to a common theme in biomedical sciences, and that is the use of guanosine triphosphate, GTP, as a timer. A large number of proteins in the cell have GTP binding and GTPase activity. That is, they bind GTP and hydrolyze it to guanosine diphosphate, GDP. And this hydrolysis event controls the function of the protein. When it is GTP bound, it does one thing. Once it is hydrolyzed GTP to GDP, it does another. And the beta-tubulin subunits are a great example of this. The GTP-bound beta-tubulin is stable and will keep the alpha-beta-tubulin monomer bound to the growing plus end of the microtubule. However, as soon as the GTPase activity of the beta-tubulin hydrolyzes the GTP to GDP, the interaction of the alpha-beta tubulins at the growing end becomes unstable and the filament starts to shrink from the positive end. Hence, if GTP levels are high, the GDP can be replaced with GTP, then the polymer continues to grow. If GTP levels are low, the filament starts to degrade. The microtubules can also be cut into smaller fragments by a protein called catenin. Like actin, microtubules can also form higher order structures in the cell. In the case of microtubules, microtubule accessory protein, also called MAPs, can bundle microtubules together by forming binding bridges between the structure and allowing adjacent microtubules to join up. Okay, so in this video we've looked at the microtubules, which is one of the filaments that makes up the cytoskeleton of the cell. We've looked at the functions of the filament, how it can be assembled and broken down, and how it can be organized in the cell.